Microfluidic systems allow rapid and sensitive detection of foodborne pathogens, such as Escherichia coli, Salmonella, and Listeria monocytogenes. These systems use lab-on-a-chip devices to perform miniaturized assays that integrate sample preparation, DNA amplification, and detection in a single platform. By leveraging small sample volumes and automated workflows, microfluidics offers real-time results, reducing the time needed for traditional culturing methods from days to hours. For example, a microfluidic device equipped with PCR technology can amplify and detect the DNA of Escherichia coli in raw meat, ensuring food safety before distribution. This application is like using a smartphone to scan and identify a barcode instantly, rather than manually inputting product details into a database. Food adulteration involves the addition of substandard or harmful substances to food products. Microfluidics enables precise detection of contaminants like dyes, preservatives, and heavy metals. Using techniques such as colorimetric assays and electrochemical sensors, these systems identify adulterants at low concentrations, ensuring compliance with regulatory standards. For example, microfluidic chips have been used to detect melamine in milk, a contaminant linked to severe health risks, by analyzing its chemical signature. This process is similar to a high-precision sieve that quickly filters out impurities from fine grains, ensuring purity. Microfluidics provides a fast and efficient way to quantify nutritional components in food, such as proteins, carbohydrates, fats, and vitamins. These systems employ fluorescence-based detection or immunoassays to measure nutrient concentrations with high sensitivity. Real-time analysis is particularly useful in monitoring food processing to maintain quality and consistency. For example, a microfluidic device can analyze vitamin C levels in fruit juices, ensuring the nutritional content meets advertised claims. This is akin to a smart thermometer that gives precise temperature readings instantly, allowing chefs to perfect their recipes. Allergen detection is critical for protecting individuals with food allergies. Microfluidic systems integrate advanced immunoassays to detect allergenic proteins such as gluten, peanuts, or shellfish. These devices enable on-site testing, reducing the risk of cross-contamination during food preparation or processing. For example, a handheld microfluidic sensor can detect traces of peanut proteins in processed foods, providing real-time safety verification. This technology acts like a personal guard dog, sniffing out hidden threats to ensure safety for vulnerable individuals. Microfluidic systems can measure biomarkers of food spoilage, such as pH changes, ammonia, or volatile organic compounds, also known as VOCs. These indicators are detected using sensors embedded in microfluidic chips, providing real-time freshness monitoring. This approach is particularly valuable for perishable items like seafood and dairy products. For example, a microfluidic freshness sensor embedded in packaging can detect spoilage markers in fish, alerting retailers or consumers when the product is no longer safe to eat. This system functions like a dashboard warning light in a car, signaling when something needs immediate attention. Microfluidics can detect antibiotic residues in meat, milk, and other animal products, ensuring compliance with food safety standards. By integrating microfluidic immunoassays with electrochemical detection, these systems provide highly sensitive analysis of residues, preventing the consumption of harmful antibiotics. For example, a microfluidic chip can detect traces of tetracycline in milk, ensuring that dairy products meet regulatory limits. This application is like using a magnifying glass to spot hidden details that would otherwise go unnoticed, ensuring thorough inspection. Microfluidic devices enable rapid quantification of microbial load in food samples, ensuring compliance with safety standards. These devices use fluorescence-based or impedance-based detection to measure the presence of bacteria, yeast, or mold. By isolating and analyzing individual microorganisms, microfluidics offers precise and reliable results. For example, a microfluidic device can quickly measure the bacterial load in raw chicken, ensuring it meets acceptable safety thresholds. This process is like a traffic counter that accurately measures the number of cars passing through a busy intersection, providing a clear picture of activity levels. Microfluidic systems are highly effective for detecting toxins produced by bacteria, such as botulinum toxin or aflatoxins. These devices integrate biosensors to identify toxins at trace levels, offering real-time results critical for preventing foodborne illnesses. For example, a microfluidic chip can detect aflatoxins in nuts or grains, preventing the distribution of contaminated products. This application is like a smoke detector, providing early warnings to prevent disasters. Microfluidics enhances quality control processes by enabling continuous monitoring of food properties during production. These systems can measure pH, viscosity, and other critical parameters in real time, ensuring that products meet consistency and quality standards. For example, a microfluidic system can monitor the viscosity of chocolate during manufacturing, ensuring it meets desired texture and taste requirements. This application is like a metronome in music, keeping everything in precise rhythm and ensuring perfect synchronization. Microfluidic systems provide rapid and precise detection of pesticide residues in fruits, vegetables, and grains. These systems use techniques such as immunoassays and electrochemical sensors to identify even trace amounts of harmful chemicals, ensuring that agricultural produce complies with safety standards. For example, microfluidic devices can detect organophosphates in apples, a class of pesticides associated with health risks, ensuring they are within safe limits. This process is like using a finely tuned instrument to detect minute impurities in a gemstone, ensuring its purity and value. Microfluidics can assess the authenticity of food products by analyzing their molecular and isotopic composition. This technology helps detect fraudulent practices like mislabeling, dilution, or substitution with inferior ingredients. For example, microfluidic systems have been used to confirm the authenticity of honey, detecting whether it has been diluted with sugar syrups. This application is like a forensic analysis that uncovers hidden evidence, ensuring the integrity of the final product. Microfluidic systems allow for real-time analysis of fermentation processes in food and beverages, providing detailed insights into microbial activity and chemical transformations. These devices monitor parameters like pH, ethanol concentration, and CO, two levels ensuring optimal fermentation conditions for consistent product quality. For example, microfluidic chips are used in brewing to monitor yeast activity, helping brewers produce beer with precise flavor profiles. This process is akin to a conductor guiding an orchestra, ensuring harmony and consistency in the performance. Microfluidics can be used to encapsulate food ingredients such as vitamins, probiotics, and flavors into microcapsules for controlled release. This technique enhances the stability and bioavailability of these compounds, allowing them to be incorporated into functional foods and beverages. For example, microfluidic systems have been used to encapsulate omega-3 fatty acids into microcapsules that protect them from oxidation while ensuring their release during digestion. This is like packaging a fragile gift in a protective box, ensuring it reaches its destination intact. Microfluidic devices can measure food texture properties such as viscosity, elasticity, and firmness with high precision. These properties are critical for consumer satisfaction and product development in industries such as dairy, bakery, and confectionery. For example, microfluidic chips are used to measure the viscosity of yogurt to ensure consistency across batches. This application is similar to a chef meticulously testing the texture of a sauce to ensure it meets the highest culinary standards. Microfluidics can optimize the delivery of nutrients and bioactive compounds in fortified foods. By precisely controlling the size and composition of nanoparticles or emulsions, these systems improve the absorption and effectiveness of nutrients. For example, microfluidic systems are used to create nanoemulsions that enhance the bioavailability of curcumin in turmeric supplements. This process is like designing a custom-built vehicle to deliver valuable cargo efficiently and intact. Microfluidic systems can perform real-time monitoring of water quality in food production, detecting contaminants like heavy metals, pesticides, and microbial pathogens. This ensures the safety and sustainability of water used in agriculture and food processing. For example, a microfluidic device can detect lead contamination in water used for irrigation, preventing its entry into the food chain. This application is like a security checkpoint that screens for potential threats before they cause harm. Microfluidics supports the analysis and customization of food designed for personalized nutrition. These systems enable precise measurement of individual nutrient requirements and create tailored solutions for specific dietary needs. For example, microfluidic devices can analyze a person's metabolic profile to recommend a personalized meal plan or dietary supplement. This is akin to a tailor creating a custom-fitted suit that perfectly matches the wearer's measurements and preferences. Microfluidic systems are being developed to monitor and optimize food packaging. These devices can detect changes in gas composition, temperature, and humidity within packaging, providing early warnings of spoilage or compromised storage conditions. For example, a microfluidic sensor embedded in vacuum-sealed meat packaging can detect oxygen levels, indicating if the seal has been broken. This application is like a thermostat that ensures a room remains at the desired temperature, preserving comfort and efficiency. Microfluidic systems can analyze lipids in food products to ensure quality and authenticity. These systems are especially useful for determining the composition of fats in oils, butter, and dairy products, as well as identifying the presence of trans fats or oxidation byproducts. For example, a microfluidic device can determine the fatty acid profile of olive oil, verifying its authenticity and quality. This is like using a spectrometer to analyze the light from a star, revealing its chemical composition with precision. Microfluidic devices allow the rapid measurement of antioxidant levels in food, providing insights into their health benefits and shelf life. These systems can perform colorimetric assays or electrochemical detection to quantify antioxidants like polyphenols or vitamin E in fruits, vegetables, and beverages. For example, microfluidics can measure the antioxidant content in green tea, ensuring consistency and quality across batches. This process is similar to checking the battery life of a device, ensuring it still has enough power to function effectively. Microfluidic platforms facilitate precise analysis of plant metabolites, enabling quality control and the development of plant-based food products. These systems can analyze secondary metabolites such as flavonoids and alkaloids, which contribute to the nutritional and therapeutic properties of plants. For example, microfluidic systems can measure flavonoid content in blueberries, ensuring they meet standards for health-focused products. This application is akin to analyzing the components of a perfume, ensuring the desired fragrance profile is achieved. Microfluidics is used to study enzymatic activity in food, such as enzymes that influence ripening, flavor, and texture. These systems provide rapid and detailed insights into enzyme kinetics, allowing producers to optimize processing conditions. For example, microfluidic devices can monitor amylase activity in bread dough, ensuring consistent starch breakdown for proper fermentation. This is similar to monitoring the gears of a clock, ensuring each one functions in harmony to keep accurate time. Microfluidic systems can analyze the physical and chemical properties of alternative proteins, such as those derived from plants, fungi, or lab-cultured meat. These devices provide detailed insights into texture, nutritional content, and digestibility, helping optimize products for consumer satisfaction. For example, microfluidics can assess the protein content and texture of plant-based meat substitutes, ensuring they mimic the qualities of real meat. This is like tuning an instrument to produce the perfect note, ensuring harmony in a symphony. Microfluidics can play a vital role in detecting and analyzing biofilms formed by microbes on food surfaces. These systems use advanced imaging and analytical techniques to study biofilm formation, structure, and resistance to cleaning processes. For example, microfluidic devices can study biofilms on produce surfaces, identifying optimal cleaning methods to reduce contamination risks. This application is like peeling layers of an onion to uncover hidden details, ensuring thorough inspection and safety. Microfluidic devices can study the interaction of emulsifiers with food ingredients, providing insights into stabilizing emulsions in processed foods. These systems can precisely control conditions to optimize the behavior of emulsifiers in dressings, sauces, and beverages. For example, microfluidics can determine the best emulsifier for maintaining the consistency of salad dressings over time. This is similar to finding the perfect adhesive to hold materials together in challenging conditions. Microfluidic systems enable detailed analysis of probiotics, providing information on their viability, metabolic activity, and interactions with other food components. These devices are critical for developing probiotic-enriched products with enhanced health benefits. For example, microfluidics can test the survivability of lactobacillus strains in yogurt during storage, ensuring they retain their health-promoting properties. This is like testing the durability of a bridge under different loads, ensuring it meets safety standards. Microfluidics can study the rheological properties of food, such as how liquids flow or how gels deform under stress. These properties are essential for ensuring the texture and sensory appeal of products like ice cream, sauces, and jellies. For example, microfluidic systems can analyze the flow behavior of chocolate, optimizing its texture for premium quality. This is akin to studying the aerodynamics of a car, refining its shape to maximize performance and efficiency. Microfluidic devices are being used to study nanomaterials in food, such as nanoparticles used to enhance flavors, textures, or shelf life. These systems provide insights into the behavior, safety, and functionality of nanomaterials in food matrices. For example, microfluidics can analyze how silicon nanoparticles interact with other food ingredients in powdered drinks, ensuring safety and functionality. This is like analyzing the performance of microscopic components in an electronic circuit, ensuring seamless operation. Microfluidic systems enable the rapid detection of mycotoxins, harmful compounds produced by fungi in grains, nuts, and other food products. These devices use immunoassays or electrochemical sensors to identify mycotoxins at trace levels, ensuring food safety and compliance with regulations. For example, a microfluidic chip can detect ochratoxin A in coffee, preventing the distribution of contaminated products. This is similar to using a smoke alarm to detect early signs of fire, ensuring timely intervention. Microfluidic platforms are used to optimize fermentation starter cultures by studying the growth dynamics and metabolic activity of microbial strains. These devices allow precise control of environmental conditions, enabling researchers to identify the best-performing cultures for specific food products. For example, microfluidic systems can analyze the fermentation performance of yeast strains used in wine production, ensuring consistent quality. This is like selecting the best athletes for a team, ensuring optimal performance. Microfluidics can analyze the microstructure of food products, such as the distribution of air bubbles in baked goods or the arrangement of fat crystals in chocolate. These insights are critical for improving texture, flavor, and stability. For example, microfluidic imaging can assess the microstructure of whipped cream, ensuring a light and consistent texture. This application is akin to examining the internal structure of a gemstone to determine its quality and value. Microfluidic devices can monitor the crystallization of sugars and salts in food products, providing insights for preventing unwanted crystallization or optimizing the texture of confections. These systems allow precise control of temperature and concentration, enabling researchers to study crystallization kinetics in real time. For example, microfluidics can be used to optimize the sugar crystallization process in candy production, ensuring smooth and uniform textures. This is similar to controlling the growth of crystals in a laboratory to create high-quality materials. Microfluidics supports the development of smart food packaging by enabling sensors that monitor environmental changes, such as gas composition or temperature fluctuations. These systems ensure that packaged food remains fresh throughout its shelf life. For example, a microfluidic sensor can detect carbon dioxide levels in modified atmosphere packaging, indicating the freshness of packaged fruits and vegetables. This is like a dashboard gauge that continuously monitors a car's engine performance, ensuring it runs efficiently. Microfluidic systems are valuable for studying the behavior of food-grade nanoparticles and their interactions with biological systems. These devices can test how nanoparticles used in food packaging or additives influence absorption, bioavailability, and safety. For example, microfluidics can evaluate the release and behavior of silver nanoparticles used as antimicrobial agents in food storage. This is akin to testing the safety and efficiency of a new pharmaceutical product before it's released to the market. Microfluidics can analyze the viscoelastic properties of dough, providing insights into its mixing, kneading, and baking behavior. These properties are critical for producing baked goods with desirable textures and structures. For example, microfluidic systems can measure the elasticity and flow properties of bread dough, helping bakers achieve consistent results. This application is like tuning the strings of a musical instrument to ensure the perfect pitch for a harmonious performance. Microfluidic devices can monitor the release of volatile compounds responsible for aroma in food products, providing a detailed analysis of flavor profiles. These systems can capture and analyze aroma compounds under controlled conditions, helping producers create consistent and appealing flavors. For example, microfluidics can be used to study the release of aroma compounds in coffee, ensuring a rich and aromatic experience for consumers. This is like using a sophisticated audio system to fine-tune the balance of sounds in a musical recording. Microfluidics can aid in studying the stability of emulsions in products like mayonnaise dressings and beverages. These systems allow researchers to observe how emulsions behave under different conditions such as temperature changes or mechanical stress. For example, microfluidic devices can test the stability of oil in water emulsions in salad dressings, ensuring they remain homogeneous over time. This is similar to stress testing a bridge to ensure it can withstand various environmental factors. Microfluidics enables rapid and precise measurement of water activity in food, a critical factor influencing microbial growth and shelf life. These systems can analyze water activity in different layers or regions of a food product, providing insights for improving preservation methods. For example, microfluidic devices can measure the water activity in dried fruits, ensuring they remain safe and stable during storage. This is like using a humidity sensor to maintain the ideal environment in a greenhouse, ensuring optimal plant growth. Microfluidic systems are used to assess protein denaturation in food products, providing insights into how processing conditions, such as heat or pH changes, affect protein structures. These devices allow real-time analysis of structural changes, helping manufacturers optimize processes to preserve nutritional and functional properties. For example, microfluidics can monitor the denaturation of whey proteins in dairy processing, ensuring consistent product quality. This is similar to monitoring the structural integrity of a building during an earthquake, ensuring it remains safe and functional. Microfluidics facilitates the study of lipid oxidation in food, a key factor affecting flavor, aroma, and shelf life. These systems enable precise measurement of oxidation markers, such as hydroperoxides and aldehydes, under controlled conditions. For example, a microfluidic chip can analyze lipid oxidation in fish oil supplements, ensuring they maintain their nutritional value and sensory appeal. This application is like using an air quality monitor to detect pollutants in real time, ensuring a healthy environment. Microfluidic devices are being used to study the thermal properties of food ingredients, such as melting points and heat transfer efficiency. These insights help optimize cooking, freezing, and other thermal processes in food production. For example, microfluidics can measure the melting behavior of fats in chocolate, ensuring smooth textures and stability. This process is akin to using a high-precision thermometer to fine-tune temperature-sensitive industrial processes. A microfluidics is valuable for the isolation and characterization of specific microbial strains used in food production, such as probiotics or starter cultures. These devices can sort and analyze microbes based on their size, shape, or metabolic activity, ensuring the selection of optimal strains. For example, microfluidic systems can isolate and evaluate specific strains of lactobacillus for use in yogurt production. This is similar to sorting gemstones by quality and size, ensuring the best materials are used for crafting jewelry. Microfluidics can analyze the behavior of food colors, providing insights into their stability and interactions with other ingredients. These systems help optimize formulations to prevent color fading or migration, ensuring visually appealing products. For example, microfluidic devices can test the stability of natural food dyes in beverages under varying pH and light conditions. This application is like testing paint colors for durability and resistance to fading, ensuring they remain vibrant over time. Microfluidics supports the development of novel food textures by studying the interaction of hydrocolloids and other texturizing agents. These systems allow precise control of mixing and gelling processes, enabling the creation of unique textures in food products. For example, microfluidics can optimize the gelation behavior of pectin in fruit-based desserts, ensuring smooth and consistent textures. This is akin to a chef experimenting with ingredients to achieve the perfect consistency in a signature dish. Microfluidics can evaluate the migration of chemical compounds such as plasticizers or adhesives from food packaging into food products. These devices provide rapid and sensitive detection of contaminants, ensuring compliance with safety regulations. For example, microfluidic systems can detect bisphenol A (BPA) migration from plastic containers into beverages, preventing exposure to harmful substances. This application is like using a metal detector to identify hidden threats in a secured area, ensuring safety and compliance. Microfluidic platforms are used to study the encapsulation and controlled release of flavors in food products. These systems provide precise control over particle size and composition, ensuring consistent flavor delivery during consumption. For example, microfluidics can create encapsulated flavor particles for chewing gum, ensuring a long-lasting taste experience. This is similar to time-release medication capsules that provide sustained effects over a prolonged period. Microfluidics enables precise analysis of sugar interactions in confectionery and baked goods. These systems study how sugars contribute to texture, browning, and moisture retention during processing. For example, a microfluidic device can analyze the caramelization behavior of sugar in candies, optimizing flavor and appearance. This process is like adjusting the balance of spices in a recipe to achieve the perfect taste and aroma. Microfluidics can study the biodegradability of food packaging materials by analyzing how they break down under different environmental conditions. These systems provide insights into the sustainability of packaging options, helping manufacturers select eco-friendly materials. For example, microfluidic devices can analyze the decomposition of bioplastics used in snack packaging, ensuring they meet environmental standards. This is akin to testing the durability of biodegradable fabrics under various weather conditions to ensure their effectiveness. Microfluidic systems are being used to analyze the interaction of food additives with other ingredients, ensuring their effectiveness and stability in various formulations. These devices enable precise control and testing of additives such as preservatives, stabilizers, and emulsifiers under different conditions. For example, microfluidic chips can test the interaction of xanthan gum with other hydrocolloids in sauces, ensuring consistent viscosity and stability. This is like testing the compatibility of different components in an electronic device to ensure seamless functionality. Microfluidics can assess the crystallization behavior of salt in food products, a critical factor for taste and texture. These systems provide real-time analysis of crystallization kinetics, helping manufacturers optimize salt particle size and distribution for enhanced sensory experience. For example, microfluidic devices can analyze how salt crystals form on potato chips, ensuring even distribution and crunchiness. This application is akin to studying the formation of snowflakes to understand their structure and uniqueness. Microfluidics allows the detailed study of starch gelatinization and retrogradation, processes that influence the texture and shelf life of food products like bread, noodles, and sauces. These systems provide insights into how temperature and moisture affect starch behavior, enabling better control over final product quality. For example, microfluidic systems can monitor the gelatinization of rice starch during cooking, ensuring an ideal texture. This is similar to monitoring the setting of concrete to achieve the desired strength and durability. Microfluidic devices are used to analyze the behavior of gases in carbonated beverages, such as the release and retention of carbon dioxide. These systems provide insights into maintaining fizz and sensory appeal over a product's shelf life. For example, microfluidics can measure the rate of carbonation loss in soda under varying storage conditions. This process is like studying the behavior of bubbles in champagne to ensure a sparkling and enjoyable experience. Microfluidics supports the development of nutrient-enriched food coatings such as edible films that enhance shelf life and nutritional value. These devices can precisely design coatings with controlled release properties for vitamins, minerals, and antioxidants. For example, microfluidic systems can create edible coatings for fruits that release ascorbic acid or vitamin C to prevent oxidation and spoilage. This application is akin to designing a protective shell for a delicate object that also provides added functionality. Microfluidic platforms are valuable for studying the moisture migration in layered or composite foods, which affects texture, flavor, and stability. These systems can analyze how moisture moves between layers in products like cakes, lasagnas, or chocolates, enabling improved formulations. For example, microfluidics can monitor moisture migration in multi-layered pastries, preventing sogginess in the crust. This is like studying water flow through soil layers to improve agricultural drainage systems. Microfluidics enables precise testing of fermentation dynamics for plant-based beverages like kombucha or kefir. These devices provide real-time analysis of microbial growth, acidity changes, and flavor compound development during fermentation. For example, microfluidic systems can monitor the production of organic acids and alcohol in kombucha, ensuring consistent taste and quality. This process is similar to a winemaker carefully monitoring fermentation to produce a fine vintage. Microfluidic devices can analyze the mechanical properties of food packaging seals, such as strength and flexibility, under varying environmental conditions. These systems ensure that packaging remains intact during transportation and storage, protecting the contents from contamination or spoilage. For example, microfluidics can test the integrity of vacuum seals in packaged meats under simulated shipping conditions. This application is like stress testing a bridge to ensure it can handle heavy loads without failure. Microfluidic systems are used to study the flow properties of molten chocolate, helping manufacturers optimize production processes like molding and enrobing. These devices provide insights into how viscosity and shear rate affect chocolate's behavior during processing. For example, microfluidics can analyze the flow of dark chocolate to ensure smooth coating on truffles. This is akin to studying the flow of lava to understand its movement and cooling dynamics. Microfluidics facilitates the study of enzymatic browning in fruits and vegetables, a key factor influencing appearance and shelf life. These systems enable precise control of conditions affecting the activity of polyphenol oxidase, the enzyme responsible for browning. For example, microfluidic devices can analyze browning inhibition methods for sliced apples, ensuring they remain visually appealing in packaging. This process is like preventing rust formation on metal surfaces to maintain their aesthetics and function.